everyone. My name is Gina Rutherford and my PhD is based upon understanding muscle dysfunction and chronic fatigue syndrome and developing a drug pre-testing system. Research has demonstrated that patients with chronic fatigue syndrome have profound and sustained acidosis with repeat exercise. This has the implication of this indicates a major muscle bioenergetic abnormality. For example, in a recent study at this university, we discovered that following a repeat exercise protocol, CFS patients basically performed a repeat exercise protocol that was 3 minutes of plantar flexion at 35% maximum voluntary contraction. And we also had a matched control group. And what we discovered was that the CFS patients had a significantly lower end exercise pH, which is indicated in the graph at the far right-hand side. Also, individual area under the curve values of pH were significantly higher. Basically, this demonstrates that the CFS patients had a combined effect of both a lower end exercise pH and also a prolonged period to recovery from acidosis. Another finding was that anaerobic threshold, which is basically the capacity, the maximum capacity of the aerobic system to produce energy before you go into your anaerobic pathway. This was significantly lower in the chronic fatigue patients, with the implication of short-term acid generation and also as an indication of an overutilisation of the lactate dehydrogenase pathway. I'll explain a little bit more about acidosis in the next slide. So basically, we found that muscle acidosis is a consequence of impaired PDC function, pyruvate dehydrogenase complex. This is a 3-enzyme complex which is involved in converting pyruvate to acetyl-CoA, which can be anaerobically metabolized. However, when PDC function is impaired, pyruvate is alternatively converted to lactic acid by a lactate dehydrogenase, resulting in an overutilization of the lactate dehydrogenase pathway, leading to acidosis within the muscle cell, and eventually an endpoint of muscle dysfunction. So what controls PDC function? Well, an enzyme called PDK, pyruvate dehydrogenase kinase, is a key modulator of PDC function. It impairs PDC function because it acts to inhibit it. However, well, that basically results in using more of this side of energy metabolism, that's how it functions. However, research has shown that DCA, dichloroacetate, which is a drug, can inhibit PDK, the enzyme. Therefore, more pyruvate is metabolized aerobically, normalizing muscle function as acidosis is reversed. The reason I've provided a bit of a background regarding acidosis in DCA is because I'll pop up later on in the presentation. Oops, sorry. So I'm now going to talk about the techniques I used in the lab to detect key analytes. So the first thing I'm going to talk about is the use of nanosensors, an intracellular measuring device. Basically, the nanosensor functions to measure analytes within the cell. The other technique I'm going to talk about is electrochemical detection, which measures analytes outside the cell. So what is the nanosensor? The nanosensor is a novel intracellular analyte detection device. It measures the fluorescent activity within the cell, and it's basically in direct real time. And basically how it functions is it's a polyacrylamide matrix. It encapsulates 2 fluorescent responsive dyes. In this case, it's FIT-C and Oregon green. And these dyes basically fluoresce with pH acid, they fluoresce. And I've got a diagram at the bottom to show a little bit more how the nanosensor works. So the analyte of interest enters the cell, enters the nanosensor cell. It then reacts with the 2 dyes, which could create an emission which can be detected by a plate reader. And the nanosensor has a number of benefits over the use of free dyes. For example, the polyacrylamide matrix prevents the nanosensor from interacting with proteins in the cell. It prevents dye leaching, and also certain dyes may have a toxic effect on cell function, so it acts as like a barrier between the cell and the analyte of interest. So how does the nanosensor get into the cell? Well, the nanosensor is delivered to the cell by a lipofectamine mediated delivery technique, which I'll show on the next slide. So basically, the nanosensors are incubated in a lipofectamine solution to form complexes. The complex then binds to the cell membrane, and the cargo is released into the cell. The nanosensors can then react with an analyte of interest. So the nanosensors can be used to detect and calculate pH change in muscle cells. However, before this can be done, a cell-free calibration curve must be produced. This is a calibration curve that I produced earlier. How did I do this? Well, basically, the nanosensors were dissolved in a buffer solution over a range of pH values, so in this case 4 to 8, which incorporates normal physiological pH. I then seeded the nanosensors into a 96-well plate, and they were interpreted using a plate reader. Once I had the output from the plate reader, I could then construct this graph, and the linear regression of this graph can then be used to be applied directly to muscle cell samples to interpret the pH value. So does evidence exist to suggest that A, the nanosensor is internalised by the cell, and also that the nanosensor is pH responsive? Well, preliminary data suggests yes to both. For example, in this cell, nanosensor doped cells were acidified with 1 micromolar of lactic acid. As you can see, after 30 seconds, the nanosensors are fluorescing within the cell. However, after 600 seconds, there has been a pH-dependent quenching in fluorescent activity. So this shows that yes, the nanosensors have made it into the cell, but also that they are responding to a change in pH. So this forms the basis for my project. Preliminary data also suggests that impaired acidosis as a consequence of impaired PDC function is completely reversible. For example, in this graph, you can see that the chronic fatigue syndrome cells exhibited significant acidosis at baseline. However, following addition of 16 micromolars of DCA, this effect is completely normalised. However, this is not the case in the non-CFS cells. So what are the implications of this data? Well, this shows that acidosis associated with chronic fatigue syndrome is reversible. However, DCA is not tolerated in the human body, it's toxic. So basically my PhD will build on this work and I'll be looking at a number of panel agents that function the same way as DCA but can be tolerated by the human body. So that essentially forms the basis of my PhD. So in addition to looking at muscle dysfunction as a consequence of acidosis, I'm also interested in looking at dysfunction through free radical overproduction. So what is a free radical? Well, a free radical is an atom that has an unpaired electron in its outermost shell. This results in a highly reactive configuration, which is demonstrated in the it reacts with other molecules to become stabilised. Superoxide is the precursor of most oxidative chain reactions. It's the product of a 1 electron reduction in molecular oxygen, and it reacts with a number of biomolecules to become stabilised, such as nitric oxide to perform peroxynitrate, and also hydroxyl radicals, which are even more damaging to the cell. Basically, this diagram shows how in excess, superoxide or other free radicals can cause damage to various points of the cell. So it may act on the mitochondria, causing damage there, which has major implications for muscle cells as they're a rich source of mitochondria. Also, it may cause DNA damage, protein membrane damage, and lipid peroxidation. So where does superoxide come from? Well, a major cellular generator of superoxide is the mitochondrial electron transport chain. Essentially, this functions by moving electrons through a chain of complexes with the ultimate aim of combining with molecular oxygen to form water, and in the process synthesizing the production of ATP or energy. However, at several stages of this chain, mainly complex 1 and complex 3, electrons may leak prematurely to combine with molecular oxygen to form superoxide. So how do I measure superoxide in my research? Well, basically I use an amperometric measurement-based technique which quantifies superoxide production following chemical stimulation in cells. It provides a real-time measurement of superoxide directly. So basically, in the presence of superoxide, I've got a little. So in the presence of superoxide, cytochrome C on the electrode surface undergoes oxidation, which is then reoxidized at the electrode surface, which generates a current which is proportional to the amount of superoxide produced. So how do I measure this in the lab? Well, this is my equipment setup. So I've got my 24-well testing plate seeded with muscle cells in each well. I submerge the probe in the well as close as possible to the cell layer but without disturbing the cells. I then use the computer software to determine a steady baseline before I stimulate it with a chemical stimulant. In my preliminary work I've been using ethanol. The reason for using ethanol is it's recognised in a number of different cellular models to be a key stimulator of superoxide production. As this forms my preliminary work, I basically just wanted to determine whether muscle cells did produce superoxide, so I used ethanol as a starting point. So what have my results shown so far? Well, my results have been quite encouraging. It's obviously very early days, but I found that ethanol has produced concentration-dependent increases in superoxide production in CFS human myoblasts. And this is also inhibited with 500 units per mil of SOD, which is an enzyme that catalyzes a dismutation of superoxide. In the next few weeks, I hope to build upon this data. I would like to repeat it in a control group to see whether